So I'm starting here with a picture of some sourdough bread. One of the things that's really interesting about sourdough bread is that most people use what's called a sourdough starter. And this starter has bacteria and yeast that really makes each loaf of sourdough bread unique. So why would a baker, someone who is using and making yeast and bacteria, need to know some biology about their bread? Well, that's what we're going to find out in this video when we talk about cellular respiration. Let's get started. So, in the last video, I introduced you to the topic of cellular respiration. Now I want to make sure we have a good understanding of the overview of everything we're going to talk about. So, cellular respiration, when you think about the word cellular respiration, right? It's the process that cells use to respire or to make ATP energy. And there are 4 stages. The first stage of cellular respiration is called anaerobic respiration. So aerobic means oxygen, anaerobic meaning no oxygen. So anaerobic respiration, the one step is glycolysis. So glycolysis takes part in the cytosol, as we learned in the last video, and that makes some ATP. You may also remember that one of the side products of glycolysis was the creation of the electron carriers NADH that are going to go over to the last step. Okay? Then we have a branching point. Pyruvate at the end of glycolysis has some options. If oxygen is present, it will go into the mitochondria for aerobic, right? Requiring oxygen. Aerobic respiration. Now, there are a few parts of that as well. The first step is pyruvate oxidation. So that pyruvate needs to become oxidized, right? We got our oxygen there. Go into the mitochondria. If no oxygen is present, then fermentation of that pyruvate will occur. And we'll talk about that in the next step. Then, so if the pyruvate had oxygen, we went in pyruvate oxidation, we will then go to the Krebs cycle, also known as citric acid cycle. And then finally, we will end with the electron transport chain and oxidative phosphorylation. So in the last video, I talked about really this step, okay? Anaerobic respiration. In this video, we want to do the rest, right? Steps 2 through 4. So I'll divide each of them up, go over pyruvate oxidation, fermentation, Krebs cycle, and finally the electron transport chain. And all of those, as you see, will be making us some ATP. Okay, so you may remember at the end of glycolysis, we're left with pyruvate. So now we have a choice. If oxygen's present, the pyruvate will go into the mitochondria, go to the next step, after pyruvate oxidation, go into the Krebs cycle and the electron transport chain. Now, if there's no oxygen present, this will lead us to fermentation. Now, there are 2 types I want to talk about, alcoholic or lactic acid. This depends on what organism you are, whether you will do alcoholic or lactic acid fermentation. So remember, glucose was broken down to pyruvate. If there's no oxygen, then we're going to have to do something else. In this case, the pyruvate went in, etc. But if there's no oxygen, look what happens. Pyruvate is converted to acetaldehyde, and then those NADH, those electron carriers we use, right, were then going to be taken back to their original NAD form, right? So what you can see here is that this process converts the pyruvate into ethanol. And you may know ethanol, of course, is what we see in our wine, our beers. Ethanol is also what we use as fuel in some cases. So this is called alcoholic fermentation or ethanol fermentation. Now we see this really in bacteria or yeast. So I started the whole video with this idea of making bread or sourdough. This is why bakers need to know biology, because this fermentation process here, right, is what is going to produce some of the byproducts, some of the CO2, some of the ethanol to make these things that we want. Okay, so If you're not a bacteria or yeast, maybe you're an animal like us, and instead of doing ethanol fermentation, you'll do what's called lactic acid fermentation. So remember the normal procedure: glucose through the process of glycolysis goes to pyruvate. If oxygen's present, it will go in here. But if there's no oxygen present, as we saw before, then we're going to go to lactic acid fermentation. So that pyruvate's converted to lactate. Same thing happens with our electron carriers. They go back to their NAD form. And then we make lactic acid. So lactic acid we see in animals. And you may be familiar with lactic acid if you do a lot of running or if you do a lot of exercise and you're not taking in a lot of oxygen, then perhaps that pyruvate will be converted to lactic acid and make your muscles feel really sore. That's that lactic acid buildup. That acid in your muscles never feels really good. Okay, so that's fermentation. Those are our two types. Now, step 2 of cellular respiration. So let's say oxygen's present. So now we're going to go, glycolysis happens. We're going to go from pyruvate in the presence of oxygen into the mitochondria. This process is called pyruvate oxidation. If oxygen's present, the pyruvate will go into the mitochondria and become oxidized. Remember, if no oxygen's present, it will go through that fermentation process we just looked at. So how does this happen? What's the reaction? So what you see is that you've got pyruvate. Here's the chemical formula. Immediately it joins something called coenzyme A. So coenzyme A joins the pyruvate, right? It comes in, we immediately make some NADH, an electron carrier, and CO2 is released. That pyruvate oxidation purpose is really to make this molecule here, acetyl coenzyme A. So you may say, why is this important, right? You see this, that was what joined here, right? And then this was what's left over from the pyruvate. So acetyl coenzyme A is important because that is what's going to enter the Krebs cycle. So the oxidation is important because only this molecule will enter the Krebs cycle in the next stage. All right, so speaking of the Krebs cycle, let's get into it. So let's review. You'll see I pop up this a lot because I want to make sure you understand where we are at each point. So we started with glycolysis, took glucose down to pyruvate. We got some ATP. Yay! We also got some NADH made, some electron carriers. Then oxygen's present, remember, the pyruvate oxidation that we just looked at, that happens. And if no oxygen, then fermentation. Well, there was oxygen in this place, so we're now ready to go to the next step, which is the Krebs cycle. Okay? This Krebs cycle will make us some ATP, but it's really going to make us the electron carriers that we need so desperately for the final step over here. All right, so let's talk the Krebs cycle. Krebs cycle is named after our friend Hans Krebs, right? Lived a long life, 81 years. And the Krebs cycle is also known as the citric acid cycle. This occurs in the matrix or the fluid of the mitochondria. Now, what happens in this step? A few important things. One, CO2 is made, right? So animals like us that undergo cellular respiration, you probably have heard, we breathe in oxygen, we breathe out CO2. This is where that CO2 that we breathe out is made. We get some ATP, and we get some coenzymes, NAD and FAD. Those will be converted to their electron carriers, NADH and FADH2. So let's remind ourselves of where this is all happening. The Krebs cycle is not really occurring on these membranes like some of our other steps, but it's occurring here in the matrix of the mitochondria, that liquid portion in the center. All right, so I'm going to give you an overview of the Krebs cycle, and then the next slide we're going to go in depth to the Krebs cycle. So remember where we're coming into. Pyruvate oxidation is this step right here. Pyruvate was oxidized, we lost the CO2, and it was converted to acetyl coenzyme A. Remember I told you this is what enters the Krebs cycle, okay? So this molecule, acetyl coenzyme A, happens to have 2 carbons. So what happens is this 2-carbon molecule joins a 4-carbon molecule already in the process. So if you take the 2 carbons and the 4 carbons, what do you get? 6 carbons. This is citrate. Citric acid cycle is where you're probably familiar with that term. So those are 6 carbon sugars, goes through the cycle. We make some CO2. Well, if I'm getting a carbon here that's released as CO2, one of these had to go away. So now that converts it to a 5-carbon sugar. Okay, so I got a 5-carbon sugar, losing some more carbon in the form of CO2. That converts it to a 4-carbon sugar. 4-carbon sugar goes through some more processes and is ready to go again. Now remember, this takes place twice for each glucose, because you may remember glucose breaks into 2 pyruvates. So this really happens twice for each glucose molecule. Okay? So this was sort of the introduction of what's happening. The next slide is a little more intense. We're going to go into a few more details, and I'm going to show you the molecules of what's happening in the Krebs cycle. Okay, so here's the cycle. Let's get going. So remember, what comes into the cycle is acetyl coenzyme A, citric acid. So you can see 1, 2, 3, 4, 5, 6 carbons because it joined a 4-carbon sugar over here. So if you take the 2 plus the 4, we've got a 6-carbon sugar, citric acid. Now that's rearranged a bit into isocitric acid. You can just sort of look, you can kind of see this oxygen here, right, has moved to there. And you can see it right there. That's isocitric acid. Now we're ready to lose a CO2. Can you look really carefully and see where that CO2 comes from? Well, it's not there. Oh, it's there. So this CO2 up there, gone. And now we have an open space here. That hydrogen and electron went to make the NADH, that electron carrier. This happens to be called alpha-ketogluteric acid. Okay? So we've lost the CO2. So notice we're down to 1, 2, 3, 4, 5 carbons in that sugar. Okay? Then we're going to lose another carbon dioxide. And if you look really carefully, you'll see that that one happens to be this one here that's on the end. Notice it's not there anymore, and we have the addition of this enzyme here, and this is called succinyl coenzyme A. So we've made some NADH, we lost some CO2, at that point. Okay, then now it's finally time where we get to make some ATP. So if you look, you'll notice in this example that I don't really see any phosphates or anything. That's because they're not labeled. But this coenzyme A is involved in the making of this ATP molecule. So we go from succinyl coenzyme A to succinate, which you see here. Notice this is a 1, 2, 3, 4 carbon sugar as well. Now our friend FAD, right, another one of our coenzymes, comes in, takes off some hydrogens from here and some electrons, and then we get fumarate. This continues around the circle. But I want to point out, FADH2 and NADH, as you see here, are our 2 electron carriers. Those are going to carry electrons over to our last step. Okay, so now we're here. We got a 4-carbon sugar. We put some H2O in. That kind of rearranges this a bit. You can see here we got an oxygen now again. That wasn't there before from here, from our water. And then as you can see here, we're going to make some NADH in a final step when this rearrangement occurs. Again, you can see this oxygen is sort of flipped up here. And that's oxaloacetate. Then the whole thing happens again. So let's do a little accounting, shall we? So remember, this all happens times 2 because we have 2 of these that come in. So I'm counting 1, 2, 3 NADHs and 1 FADH2. But remember, this is all multiplied by 2. So I'm really getting 2 FADHs, 8 NADHs, 2 ATPs. All right, so we see our 2 ATPs, but 1, 2, 3, I'm only getting like 6 NADHs. So remember, because this acetyl coenzyme A that happened during pyruvate oxidation, that also made an NADH. Okay, so really, the Krebs cycle, including pyruvate oxidation, gives us 2 ATPs, 8 NADHs, 2 FADH2s, and 6 CO2s. And you may say the same thing. Well, I'm only getting 4 CO2s here, but remember that other CO2 was also in pyruvate oxidation. Okay, so here's your energy accounting if you want to account for all of the energy and all of the products that we are seeing from the Krebs cycle. Okay, finally, stage 4. Stage 4 of cellular respiration is what we call the electron transport chain and oxidative phosphorylation. Well, let's think about this word. Oxidative, you may remember, I told you Leo the lion says grr. When we lose electrons, that's oxidized. When we gain electrons, that's reduced. So if oxidation's occurring, something must be losing electrons, okay? And then the last part of this, phosphorylation, means something's getting a phosphate group. So we're losing electrons and getting a phosphate group in this last step. So let's review it. Glycolysis takes glucose down to pyruvate. We get some ATP. We also get our friend, the electron carrier, NADH. If oxygen's present, that pyruvate, of course, will go into the mitochondria through the process of pyruvate oxidation. If no oxygen's present, fermentation occurs. We talked about the 2 types, alcoholic and lactic acid. Now, after pyruvate oxidation, that will go through the Krebs cycle, what we just went over in great detail. That will make some ATP, but also some NADH and FADH2. You might want to add that as well. So we are finally ready for step 4, the electron transport chain and oxidative phosphorylation, where we're going to get a lot of ATP. So the electron transport chain is lots more ATP, giving us a lot of ATP. So let's talk about where this occurs. So here's our mitochondria, powerhouse of the cell, ATP maker of the cell. You see the outer membrane and the inner membrane. So we're really in this spot here, outer membrane and an inner membrane. So we're kind of in this space where we're going to see the electron transport chain take place. So we're going to use the electrons that are brought over from NADH and FADH2, right? Those are our electron carriers. Those electrons are going to establish an electrochemical gradient, right? So there's a higher percentage of something on one side than the other. There's a gradient. That's going to be established. And per 1 glucose molecule, we're roughly going to get about 34 ATP in this process. Now, you remember, this only occurs in the presence of oxygen, so it's an aerobic process. And we see it occurring in the mitochondria. So let's get into it. Here's the electron transport chain. These are the steps. I'll go through them, but I think it's easier to just show you on the diagram. So remember where we are. Here's the mitochondria. Here's the outer membrane, inner membrane, the space in between them, intermembrane space, and then this fluid inside that you see all in here is the matrix. So let's see what's going on. All right, so here's the electron transport chain. Remember what's coming in. It starts with this NADH, our electron carriers carrying in our electrons. FADH2 also carrying some of our electrons. So when they get here, notice what happens. Those electrons are cleaved off. So here's my electrons now. That leaves me with NAD and some hydrogens. Same thing happens to FADH2. Electrons cleaved off. That leaves me with FAD and some hydrogens. Well, what are those electrons are going to do now? Well, they need to go through the chain. That's why we call this an electron transport chain, because they're going to go through this chain of proteins, and as they go, those electrons, they're going to start here, and then they're going to fire through here, yay, zzz, all the way through that electron transport chain. What that does is that provides the opportunity, right? The electrons, you might think of it as sort of like an electrical flow, electrons. When those go through here, that takes the hydrogen and shuttles them across. So we've got these hydrogens that were here are now going to be shuttled across. Hydrogens up here, right? So now we've got all these hydrogen ions, these hydrogen ions up here. Same thing goes on with FADH2. Those hydrogens are lost. They're moved across this chain because the electrons are activating these proteins so that the hydrogen atoms can be shuttled through. So this process is the electron transport chain because the electrons are being transported through this chain of proteins. Now, what happens to this electron when it gets down here, right? So the electrons, here they move through the chain. When they get done with this last protein in the chain, they need to go somewhere because electrons can't just be hanging out, right? We don't want random electrons. So what happens is some oxygen binds to the electrons with hydrogen. So we say that oxygen is the electron acceptor. So oxygen accepts those electrons, binds with some hydrogen to make water. Did you know that your mitochondria actually made water for you? Right? Pretty interesting. All right. So what's the point of all of this? The point is that now we have all of these hydrogens up here. So we have a gradient, right? Where's my word gradient? Well, it's not up here, so let me write it. We now have a gradient of a high concentration of hydrogens here and a low concentration of hydrogens there. So anytime we have high concentration on one side of a membrane, they want to go from high to low. So they find an enzyme, ATP synthase. This enzyme does what it says. It synthesizes ATP. So the hydrogens start going through. They create what we call a proton motive force. Essentially, if you think of a hydrogen it has one proton, so we could just call it a proton. So a proton motive force takes ATP synthase, turns it on, gets it really fired up. That process then takes ADP plus a phosphate and makes it to ATP. Okay? So we need this enzyme turned on to take ADP plus a phosphate to make ATP. This step here is called oxidative phosphorylation. Why? As I mentioned, because this oxidative process of losing the electrons going through the chain creates this phosphorylation, or the phosphate group being added to the ADP to make ATP. Okay? So I know that's a lot. This is complicated. But what you need to remember, bottom line, ATP is made because this electron transport chain shuttled across hydrogens, creating this gradient which drove or turned on ATP synthase. All right, so speaking of ATP synthase, what is it? Well, it ends in ASE, so you know if you're a good biology student that that's an enzyme. Located in the inner membrane, and it basically takes ADP plus a phosphate to make ATP. So adenosine diphosphate, 2, plus a phosphate, 1, gives us adenosine triphosphate, 3 phosphates. So this proton motive force, as I mentioned, creates this concentration that turns it on. Now, this is a very complicated enzyme. There's actually like a rotor, and this thing kind of spins. And that action actually creates the space for ADP to add that phosphate group to make ATP. Okay? ATP synthase, very important enzyme. It gives you the energy you need for your body to work. Now, I'm giving you a random picture I found on the internet of a different overview of cellular respiration. I don't want you just to memorize my diagram. It's better that you have a full understanding and that you can interpret different diagrams. So here's a random one I found on the internet that was royalty-free. So this is just a different view. I'm going to walk you through it and then we'll answer some questions to close up the video. So remember, the cellular respiration process. Let's remember the formula. C6H12O6 plus oxygen is going to give us carbon dioxide plus water plus ATP. Now, remember what we start with? Glucose. So here it is. Glucose, there's our molecule, goes through glycolysis. I like to use a Y there. I think that's maybe that's the British way you say it. I don't know. Glycolysis with a Y takes glucose, breaks it down into 2 pyruvic acids, and remember we get some ATP. If oxygen's present, that goes through pyruvate oxidation, and it's converted to acetyl coenzyme A to go into the Krebs cycle, citric acid cycle. Remember, glycolysis gives us some NADH. You see that line there? Pyruvate oxidation gave us some CO2, as well as the citric acid cycle gave us some CO2 as well. Now, electron carriers are what's made in citric acid cycle, NADH, FADH2. All of those electrons are then going to go over to this final step, electron transport chain, where those electrons go through these proteins, right, to make the electron transport chain activate, which will take the hydrogens, shuttle them across so that it builds up a gradient. That gradient then will go through ATP synthase, making a lot of ATP. So here's the summary of cellular respiration. What I want you to do is I want you to go through these questions, pause the video, see if you can answer them on your own, and then I'll be back to answer them and help you make sure you've got the right answer. Okay, so let's go over these one by one now. Here's the equation. Where did the glucose come from? Okay, well, hopefully you got that glucose from eating something, right? The glucose came into your body, you broke down some carbs. Where did the oxygen come from? Well, hopefully you breathed that in and it came into your cells. Where did the CO2 come from? Remember, the CO2 came from the cycle, the Krebs cycle, right? So that came from the Krebs cycle, or the citric acid cycle. We also made some CO2, if you're getting really specific, in pyruvate oxidation. Okay, where did the water come from? Oh, remember, we made the water when oxygen accepted that electron and joined with hydrogen to create water in the electron transport chain. Where did the ATP come from? Came from several places. Glycolysis, Krebs cycle, but the majority of it came from the electron transport chain and oxidative phosphorylation. So why do we breathe? Oh, well, the quick and easy answer is if we didn't have oxygen, our pyruvate would not be able to go into the Krebs cycle to make ATP, and instead would go through fermentation and we would not survive, right? Because we need that ATP to be made. So hopefully this video helped you understand a little bit more about this very complicated process, cellular respiration, how our body, how the mitochondria help to make energy in the form of ATP.